Good morning. This is Miss Dagenford. We're going to be looking at red blood cells today and the hemoglobin that is found within them. Some basic information about them. They are about 7.5 micrometers, by maybe 1.5 or 2 micrometers. Now, the take-home message for them is that they are very small. That's the idea. You might recall earlier in the year, we looked at these under the microscope, and they really do have that kind of lifesaver appearance, and that is due to the fact that these are biconcave, and the biconcave characteristic is seen in the middle where the red blood cell just appears to be squished a bit, and this biconcave gives it just that little bit more surface area to help oxygen be absorbed. The other notable characteristics is that it has no nucleus. And this is going to be important because without a nucleus, it cannot perform any transcription. There is no more DNA. And without that DNA, there's no more ability to make new proteins. Now, as long as there are mRNA molecules in that cell, it can still perform protein synthesis, but that's only with the mRNA that has been made. The other fact that is pretty unusual is that they eject most of their mitochondria. And mitochondria, if you recall, the mitochondria are there to complete cell respiration. And so if we did a little bit of a sneak back to earlier in the year, you'll recall that we have glycolysis, and that happens in the cytoplasm. And that only makes 2 ATP, and there is no oxygen needed for this. But then the glucose that is broken down into pyruvate, that pyruvate will then enter into the mitochondria. And this is why we see the absence of mitochondria, because what will happen in the mitochondria will use oxygen to complete the oxidation of that pyruvate. So even though we might be able to get that wonderful 28 to 30 ATP from the rest of this process, the problem is it requires oxygen, and that would consume the oxygen that the red blood cell is carrying. So without mitochondria, it will not consume the oxygen that it carries. You might recall, if you were in honors anatomy, you would recall that I used the analogy of a donut delivery dude, and the last thing we want our donut delivery dude to do is eat the donuts he's carrying. And so just as the donut delivery dude does not eat the donuts he's carrying, we do not want the red blood cell to consume the oxygen it's carrying. But you may ask, well, does it not need ATP? Well, it can still undergo glycolysis. It still has all of the machinery to do the glycolysis. And so it can get that little bit of 2 ATP from glycolysis. And they're not going to be the most metabolically active cells anyway. They really are just carriers. They are storage. They also don't have most of the organelles. Um, so we want to get rid of the all of the organelles that take up space. Now, why do we want to get space? Well, if we scroll down here, we want to get space for hemoglobin. Hemoglobin is going to be the important molecule here, and you'll notice, let me zoom in a little bit because it's hard to see on mine. Hemoglobin has this very interesting shape. It has four polypeptides. These are the four globin polypeptides. And this would be a good example of a quaternary structure of a protein where you have multiple polypeptides. And the globin structures, each one contains a heme group. Now we have two alpha, that's alpha, A-L-P-H-A. We have two alpha proteins and we have two beta globin molecules. And these are going to be the ones that contain the heme groups. And you'll notice over here we have one heme group. There's a second one, there's a third one, there's our fourth one. So there you can see the heme groups kind of nestled within those globin protein molecules. What's going to be important here is what's inside the heme groups. That's going to be iron, and the iron is going to bind to the oxygen. And when we have hemoglobin, which is abbreviated HB, when we have hemoglobin plus oxygen, we will call it oxyhemoglobin. And when it's without oxygen, we would say deoxyhemoglobin. And the other thing that we do see with hemoglobin is something interesting called cooperative binding, where once we see one oxygen, the following oxygens that get bound, it becomes easier. So once one oxygen becomes bound to a heme group, the other heme groups bind to the other oxygens far easier. And faster. So we call that the cooperative binding, and you see that down here, cooperative binding, cooperative affinity. So if we were to try to draw it, here's one globin, here's another, here's another, here's another. If we were to kind of indicate, okay, here's going to be one oxygen that binds inside that heme group. Well, once that oxygen binds, the other ones bind much faster. And that's called cooperative binding, cooperative affinity, and that has a lot to do with how the shape changes as the oxygen binds to it. It affects the affinity for the other globin molecules in the heme groups for the oxygen. This happens in reverse too. So when it loads oxygen, it happens faster. But also when it unloads oxygen, it happens faster. So oxygen unloading, once one oxygen leaves, others leave faster. And that's going to come into play as we look at our next topic, which is going to be the oxygen hemoglobin dissociation curve. Well, thank you so much, and we will talk later.